Yo, what's up, everybody? I got another review here that I wanted to do. Didn't get a lot of questions about muscle fizz. I'm assuming that, the notes were pretty good and the videos were pretty good too. I just wanted to talk about a couple of things. Well, okay, I'm lying. I want to talk about more than a couple of things. I want to actually talk about several things and try to get your mind working about muscle physiology because muscle physiology can be one of those things where people assume that they understand what everything means, and then they get into the test and then they find out, oh man, this thing was horrible. Looks like I didn't understand this stuff as much as I thought I did. So I kind of want to pick at your brain a little bit, see what you know, see how that flows. So let me go on over to the screen share and we'll get started. I want to take some things from some of the notes and maybe see what we can see. And I'll get to the screen share here in just a moment. And then we'll pull up the PowerPoint. I kind of want to start with slide 71 and kind of talk about some things there. When you're talking about this slide, we're talking about the neuromuscular junction and everything that's happening here. And you need to come to an understanding that the neuromuscular junction is nothing more than where the motor neuron, that nerve cell, and a muscle cell, it's where the communication actually takes place. It's where the handoff happens. Your motor neuron, that nerve cell, is now communicating with your muscle cell. This is what's going on. So there has to be the start and the finish of this. The motor neuron is receiving signals from the nervous system, more importantly, the central nervous system. So at some point in time, somewhere in your central nervous system, either your brain or your spinal cord, you have created a motor command and you're sending it out through this motor neuron. And that's where that term nerve signal happens. If you look up there where it says nerve signal here, and you're looking at that little orange arrow, that nerve signal is that action potential that's traveling down the cell membrane of the motor neuron. It's an electrical current. It's a little millivoltage electrical current that's traveling down, and when it gets to the very end of this motor neuron, down here where the synaptic knob happens to be, that electrical current causes voltage-gated channels to open on the surface of the synaptic knob. And those voltage-gated channels, these are channels or doors that are controlled by an electrical current. Just like when you're going to Target or Walmart or some grocery store, Publix, Bilo, wherever you're going, when you go to these stores, you'll notice that the doors open automatically. There are electrical pulleys, little motors that slide the doors open so that you can walk through. That's what a voltage-gated channel happens to be. It's an electrically controlled door that only opens when an electrical current comes into contact with the door. And when these doors open, they are specifically designed to allow calcium to come out of the intercellular fluid. The extracellular fluid and into the actual cell. In this case, you're seeing calcium float around the outside of the cell here, and then the calcium slowly moves its way in through the pump into the cytoplasm inside of the synaptic knob, and it moves in through basic diffusion. So you got this high concentration of calcium that moves in to this area of low concentration of calcium, which is inside the cell. The calcium comes in, and then the calcium attaches itself to the synaptic vesicles. When it attaches itself to the synaptic vesicles, this leads those membrane-bound bubbles, those synaptic vesicles, those trash bags filled with neurotransmitters. The calcium stimulates those big bubbles to move towards the edge of the synaptic knob, fuse with the synaptic knob, and then dump its contents out into the synaptic cleft. Now, someone asked me a question beforehand, and it was a great question. They were talking about does the calcium go into the synaptic vesicles? No, it does not. the calcium is not becoming anything here. The calcium remains calcium. The only reason why calcium is on the scene is so that calcium can cause the synaptic vesicles to release their contents into the synaptic cleft. And then what happens to the calcium? Well, the calcium stays inside the cell and it ultimately gets pumped back outside of the cell at some point in time. But that's not what's important right now. What's important is the fact that the calcium came in. And once it came in, it stimulated the synaptic vesicles to move out and then dump its neurotransmitters out into the synaptic cleft. And the name of these particular neurotransmitters happen to be acetylcholine, which is written in shorthand ACH. Now, this is what's happening in a neurotransmitter, in a neuromuscular junction between a motor neuron and a skeletal muscle cell. If we look at a synaptic cleft in your brain, the neurotransmitter being released may not be acetylcholine. The neurotransmitter that's released may actually wind up being, something like nicotine or dopamine. It may be GABA. It may be in any number of different neurotransmitters. There's like, I could sit here and name somewhere around 50 different chemicals that your brain uses as a neurotransmitter. In this case, we're looking at the motor neuron talking to the muscle cell, and it's using acetylcholine as its communication. If you were growing up in school like I did, then you know about passing notes in class. And so someone passes you a note in class. As a matter of fact, I teach college and I still have adults pass notes in class. I have adults in my class who are older than me who pass notes in class right there in front of everybody. So, this is what's actually happening here. The motor neuron is passing a note in class to the motor cell, to the muscle cell, to get the muscle cell to do something. And that something that he wants him to do is to contract. That is the main goal, is to get that muscle cell to shorten, to contract, to shorten itself, to ultimately get the mechanisms inside of the muscle cell to activate, to do their job, to do what they do, and then cause that muscle cell to shorten, therefore pull and therefore pull and create a pulling motion or create tension, which therefore leads you to be able to move. Notice that the acetylcholine in this image here in the synaptic cleft lands on these things, these ACH receptors, which happen to be bound to the motor end plate, which the motor end plate is a specialized section of the sarcolemma, or the cell membrane of the muscle cell, designated to catching and receiving acetylcholine. That's why you have a motor end plate. Think of a motor end plate as like a landing pad. When you see a helicopter go to land somewhere at a hospital, the helicopter does not try to land in the parking lot. The helicopter does not try to land in the middle of a grassy field. The helicopter has a landing pad that it would like to aim for and that it would like to land on, and everyone's waiting near that landing pad, waiting on that helicopter to bring that patient in or pick that patient up. So the same thing happens here. The motor end plate happens to be a landing pad where the acetylcholine can attach to the binding sites on these receptors, and then these channels open up. See the little blue things down here on the motor end plate? These big channels open up. These channels are a lot like these channels, except for one major thing. These channels up here on the synaptic knob are voltage gated channels, so they open in the presence of the right voltage, of the right electrical current. These channels down here, these big blue ones, they are not voltage gated channels. They are chemically gated channels. They open when the right chemical is exposed to them. And then they open and they allow sodium ions, which are not shown in this picture. Sodium ions rush in, and then they create an electrical current on the surface of the muscle cell, which is going to lead to the mechanisms inside the muscle cell to activate and create a muscle contraction. Now, with that being said, I want to move on to something else. I feel like my slides are off in their numbers. This is kind of crazy. Maybe I need to go backwards. Yeah, needed to go backwards. Let's go back to slide 66, and we see here this is the overview of events in skeletal muscle contraction. You know, if anybody were to take a test on this stuff, you want to be able to know this picture by heart, and you want to be able to express all the details of what you see in this image. I thought that what they did with this image was pretty cool, where they said, okay, we're going to break this whole process down into three unique events. And there's a lot of subplots in it. Just kind of think of it like Game of Thrones when you buy the box set of Game of Thrones, or if you buy the Lord of the Rings box set, and you know how you have the Lord of the Rings, the Two Towers, Fellowship of the Ring, and then Return of the King. You know, you got all those little subplots and all those little sub events that happen, but you summarize it all up on these three compilations, these three movies. So in movie number one, it's got the box at the top. That's neuromuscular junction, excitation of skeletal muscle fibers. We talked about this. We just finished discussing how an action potential, that electrical current, comes down the axon, gets to the synaptic knob, stimulates the voltage-gated channels to open on the synaptic knob, therefore leading the calcium to flood into the synaptic knob. The calcium comes into contact with the synaptic vesicles. The synaptic vesicles move towards the edge of the synaptic knob. They release their contents, acetylcholine, into the synaptic cleft. The acetylcholine binds to binding sites on the surface of the motor end plate, aka the specialized landing pad on the sarcolemma, also known as the cell membrane of the muscle cell, and that generates an action potential, which leads to step two, which movie number two shows you the sarcolemma, the T-tubules, the sarcoplasmic reticulum, also known as the SR. And what's really happening here is the action potential at the time that you see it here between 1 and 2, the action potential is traveling across the surface of the muscle cell, but it hasn't gone in. And because it hasn't gone into the cell, ain't nothing going to happen yet, because the action potential is on the outside, and therefore if the action potential stayed on the outside, it would only stimulate the myofibrils that are closest to the surface. Therefore, all of our muscle contractions would be uber weak, and you wouldn't be able to pick up your book bag to even go anywhere. So what winds up happening is we find out we got to get this action potential inside of the cell where it can hit all those myofibrils that you see wrapped in there looking like a big cigarette pack. So what winds up happening is we have T-tubules, which are these little tubes or invaginations into the cell, which is a fancy way for saying we've got some Teenage Mutant Ninja Turtle sewer pipes that run from the surface of the cell membrane down into the depths of the actual muscle cell so that it can link that action potential, that electrical current, with every single myofibril. That's what it means. So if you look at the green thing in this picture, you see the orange arrow. Notice that the orange arrow travels, the action potential travels across the sarcolemma, across the surface, and then all of a sudden it goes down in that green tube, and it's going down that green tube so it can get inside the actual muscle cell and therefore stimulate the sarcoplasmic reticulum, which in this case, the sarcoplasmic reticulum is the squiggly blue stuff. And the squiggly blue stuff is therefore stimulated by that same electrical current. And the squiggly blue stuff has a primary job, which is to store calcium ions. Once again, we see calcium ions show up on the scene and do us a favor of stimulating some type of activity. So first we saw calcium ions show up in the motor neuron, that nerve cell, and make something magical happen. Now we're seeing the calcium show up in the muscle cell, and now it's going to make something magical happen. It's not the same calcium that you saw before. This is a different storehouse of calcium. Calcium is calcium, you know, it's all the same, but it's coming from two different places. The calcium that was helping the motor neuron came from outside of the motor neuron. The calcium that we're seeing here was actually being stored in the muscle cell, inside the muscle fiber. So when the action potential travels down the T-tubule, because the T-tubule and the sarcoplasmic reticulum are physically touching one another, that electrical current, that action potential, stimulates the SR, the sarcoplasmic reticulum, to release its calcium. And when the calcium is released, The calcium now moves on into movie number 3, the finale of the trilogy. And that's when the calcium is now floating around inside the muscle cell, and it comes into contact with something called troponin, which, when it comes into contact with troponin, it therefore causes an interaction between troponin and tropomyosin. Now, this is where I think that things get kind of convoluted and kind of twisted and kind of confusing. And so this is where I want to slow down just for a moment. There are 4 protein fibers that you really need to concern yourself with. I know that someone would tell you there's more. Guess what? That's not what we're going to talk about. We just want to talk about these 4 today. That's troponin, tropomyosin, actin, and myosin. All right. Now, the 4 of them are in a relationship with one another. All right. There's troponin, tropomyosin, actin, and myosin. Troponin, tropomyosin, actin, and myosin. This is the relationship. Actin and myosin are supposed to be hooking up. Myosin is supposed to grab actin and pull actin so that the actin fibers come together, cause the muscle cell to overall shorten, and therefore cause a contraction. That's what we ultimately want to happen. However, to keep your actin and your myosin from acting up and hooking up with one another anytime they want to, what we wind up having to do is we have to put binding blockers on actin so that myosin can't just randomly grab a hold of actin and pull actin anytime it wants to. This is very important. Otherwise, we would have actin and myosin grabbing one another, contracting, and you would have your muscles contracting, and your body would be moving and doing some stuff that you really had no business doing. Because, you know, if your actin and your myosin started contracting with one another while you were on a crowded elevator, there'd be some angry people up on that joint. I can just, you know, say that for sure. So in order for our myosin and our actin not to act up, we have troponin and tropomyosin in place. Troponin and tropomyosin are the security guards for actin. They make sure that myosin cannot get to actin. Troponin and tropomyosin are player haters. They wrap around actin and keep actin covered so that myosin cannot bind to it, therefore dragging the thick and thin filaments together and therefore creating a contraction and causing a very awkward situation for you on that elevator. So troponin and tropomyosin, these guys, these are the security detail. They are the security guards for actin, and they block access to actin. So when calcium is released, calcium binds to troponin, which then forces tropomyosin to move, exposing binding sites on actin and making actin a free target for myosin to reach out and attach to. So it's kind of like troponin and tropomyosin are the security guards for actin, but calcium is kind of like a payoff. It's a bribe. And so you bribe troponin, who then talks tropomyosin to move out of the way, and then that's when myosin can now attach to actin and cause the thick and thin filaments to draw together, therefore causing a muscle contraction. This is when the second law of ludicrous comes into play, where troponin and tropomyosin cause themselves to move and get out the way so that myosin can attach to actin. Yeah, I said it. And yes, it's recorded. And some of you probably didn't catch what I just said, but for those of you who are hip hop lyrically challenged, just YouTube "Move, Get Out the Way" by Ludacris, and you'll figure out what I'm talking about. I will not be held accountable for the other lyrics that you may hear in that song, so please don't hate on the brother. But this is what's happening. The tropomyosin and the troponin have a relationship, and the actin and the myosin have a relationship as well. So that handles those 3 boxes right there. If you followed what I just said and it was like, oh yeah, that was peachy keen, I figured that out, we're good, then we're fine. We are perfectly fine. But if what I was saying left you lost, you need to rewind this thing and go back and look at this image again. Pull out your notes, go back and look at the videos that we were looking at, the animations. Maybe we can describe that a little more in more detail later on as well. So I said we are going to talk a little bit about some pathology, and that we will. I want to keep my promise on that. I actually want to talk about myasthenia gravis, and I want to talk about um, botulism, and I might talk about a third disorder. It depends. We'll see how this goes. Let's talk a little bit about myasthenia gravis. Myasthenia gravis is an autoimmune disease. So, you know, they talk about how it's primarily in women, but if you Google myasthenia gravis, the first picture that's probably going to come up is a picture of a guy. He was a very famous case of myasthenia gravis. So don't be alarmed by that. Myasthenia gravis, when we say it's an autoimmune disease, when you say something's autoimmune, basically you're saying that the body is attacking itself. The body is having trouble recognizing itself. We've got friendly fire taking place. We're firing on ourselves. We're attacking ourselves, and we're destroying ourselves. So you can see here in the second line, it says that antibodies binding to acetylcholine receptors in the neuromuscular junctions, and the receptors are removed from the muscle fiber by endocytosis. Now, this is a big fancy way of saying this. You know, when you go to attack something in your body system, generally you attack it because you don't recognize it. That's why your immune system attacks something. Your immune system. Anybody ever saw the old school movie? What's that old movie? There's an old movie, Osmosis Jones, and it's about the white blood cell who's a detective, and he's trying to hunt down the evil virus and all that. You know, that was a really good movie. There were a lot of things in that movie that you could actually use to teach immunology from. The only problem with that movie, first of all, is that, you know, all the cells and all the microorganisms in that movie could see, and your the cells that make up your specific immune processes, those cells don't have eyes. They can't see what's wrong. So everything looks alike to them because they can't see anything anyway. So it's not like they can tell a diseased, or I shouldn't say a disease, of it's not like they can tell a cell that's been infected by a virus from a normal cell. They can't tell a normal cell from an abnormal cell just by looking at it, but they can tell a abnormal cell from a normal cell based on protein receptors that are located on the cell membrane. Now, I'm going all the way around the world on this to explain something key, because if you're going to take anatomy and physiology, you're going to have to understand all of this stuff. So you might want to get it now. So what these immune cells do is they simply scan your body cells on a day-to-day basis, basically looking at these little receptors on the cell membrane, just like a state trooper looks at your license plate and looks at the tag on your car to make sure that you are who you say you are, and to make sure that car isn't a stolen car, make sure that's not an abnormal car, and that is a normal car. Your immune system does the exact same thing with all of your cells, but in an autoimmune disease, the body fails to realize that even though it's the body of California and that those are California license plates, it treats those California license plates like they're New Jersey license plates, and it pulls it over. And so in this case, what winds up happening is, you remember those acetylcholine receptors that you find on the motor end plate? Right there in the synaptic cleft? Ah, yeah, you forgot about that just that quick, didn't you? You need to go back and take a look at that. You got to remember. Those acetylcholine receptors that are on the surface of the motor end plate, on the muscle cell, those receptors, for some strange reason, the immune system does not recognize them, and therefore it creates antibodies to bind to them and mark them for deletion. Literally, the antibodies bind to them and mark those receptors to be destroyed. Well, if I destroy, I want you to put some thought into this, okay? If I destroy the receptor sites on a motor end plate in a neuromuscular junction, then the acetylcholine doesn't have anything to bind to. If most of the receptors have been destroyed, then the acetylcholine shows up, and only a few acetylcholine molecules actually get a chance to bind to anything. Therefore, when they go to bind to the few remaining receptors, that makes the action potential that's created on the muscle cell very weak. And if the action potential that it just created is very weak, then that whole process inside of the muscle cell, remember the second and third movie? Halfway through the second movie and the third movie and the picture we were just looking at, that means that those processes are going to be extremely weak as well, which means that the muscle cells are going to be very weak, which means that you're not going to have much strength to be able to do much of anything, which is going to put that person into possibly, an automated chair or something if it hits their legs. And it means that their muscle cells are going to fatigue really easy because you're unable to maximize the amount of myofibrils inside of the cell that you can use, because the action potential that you sent down there is so weak, you're only stimulating a handful of a handful of mechanisms inside the muscle cell, whereas you needed to create enough tension to actually stand up and walk across the room and pick up your suitcase and walk out the door. Instead, you're only able to produce enough tension to like barely move your leg and wobble over to the wheelchair and sit down in it. One of the first places involved with myasthenia gravis is around the face. It usually attacks facial muscles. That's why when you look at if you Google myasthenia gravis and you look at images, you'll notice that one of the similarities that people share is they have the droopy eyes and the drooping face. Where the muscles in their faces and around their eyes, those muscles have relaxed extremely, and so they're relaxed to the point where they're not contracting or causing anything solid to happen. No solid controlled motor movements. If we skip along a while, I wanted to take us to another disorder. Talk about tetanus just a little bit. Tetanus is what we call a form of spastic paralysis, where this can be life-threatening. All right, tetanus is the whole reason why we wound up creating the tetanus shot. It's actually caused by the toxin of a bacterium known as Clostridium tetani. Now, it's not, I want to say this, and I got to be careful how I say this because this can kind of get you in trouble. It's not the bacteria that's the problem. It's the toxin created by the bacteria that's the problem. If you took the bacterium and you laid the bacterium on the side of your muscle and said, here, muscle, let me introduce you to the bacterium, there wouldn't be much of a problem. But once that bacterium starts producing that toxin, now we got a problem. And it produces toxins just like you produce sebum, or earwax, or sweat, or tears, or saliva in your mouth. I mean, toxins are usually, not always, but depending on the bacterium, sometimes toxins are nothing more than a waste product that the bacterium is creating while it's metabolizing certain substances. So this toxin is produced by this bacterium, and the reason why this toxin is so dangerous to people is because the toxin blocks the release of the inhibitory transmitters in the spinal cord. All right. Now, what I mean by that is this: your spinal cord has to release inhibitory neurotransmitters so that you stop sending motor signals to certain muscles. All right. As long as there's no signal coming through the motor neuron, your muscle cells are not going to contract. You're good. If everything's normal, you shouldn't have anything to worry about. But if I block those inhibitory neurotransmitters, in other words, there are some receptor sites that the toxin goes in, and the toxin binds those receptor sites. And so now you cannot, the inhibitory neurotransmitter chemicals cannot bind to those same sites to allow you to be able to stop contracting your muscles. I was watching a movie one time, and it was an action thriller. and the girl was running from the killer. And I was trying to figure out how this girl was going to get away from this killer, because there was no way she was going to be able to fight back against this dude. He was going to wax her for sure. And so what she did was she went to open this door. She had a key, and the killer was coming for her. And she had the key, and she locked the door. But after she locked the door, she broke the key off in the keyhole and then ran. And so when the villain got there right behind her, he goes to unlock the door, and then he says, "Oh, well, I'll just use the key." Oh, yeah, chick was smart. She broke the key off in the keyhole. Ah, she's a smart cookie. Well, yeah, so the villain couldn't get through the door, and she got away, and she wound up hooking back up with the hero. That was great. Unfortunately, here we got the reverse. Instead of the hero and the heroine pulling this trick, we got the villain pulling this trick. So we've got the villain, who is Clostridium tetani, releasing a toxin, and the toxin binds to the keyhole, that binding site where the inhibitory neurotransmitter key was supposed to go in to help your muscles be able to relax. So now that keyhole is full of the toxin, and you can't put an inhibitory transmitter there to tell your muscles to relax. So what winds up happening is you have an overstimulation of the muscles, and this can be life-threatening, because what the average person doesn't know is that the muscles that help you breathe are skeletal muscle. Yeah, the same muscles that help you walk and lift a fork of pecan pie to eat, that same kind of muscle is the same kind of muscle that helps you breathe, like what you're doing right now. Like, your diaphragm is responsible for 75% of the air that you breathe in. It's responsible for 75% of inhalation. So if that toxin by Clostridium tetani hit your diaphragm, how would you breathe? You'd only be able to breathe using your accessory muscles and a few intercostal muscles. Breathing would be extremely difficult. Your muscles would fatigue after a while, and you would stop breathing and you die. Botulism. You've probably heard of botulism. Anybody who's been in this country for a while, you've seen some botulism outbreaks in the last few years. It's been ridiculous. There was this dude up north who got fired from a cabbage and collard green canning plant, and to exact his revenge, he decided to dump a little bit of fecal matter in one of the main vats that contain collard greens, and they had to shut the whole thing down, and they had to recall all of those canned collard greens, and they had to hunt that guy down too, and he got locked up for it. And the reason why he was doing that was because he wanted to spread the bacterium Clostridium botulinum. Clostridium botulinum is another bacterium, and it produces a toxin that actually prevents the release of acetylcholine at the synaptic knobs. And so what winds up happening is you can't get acetylcholine across the synaptic cleft to actually bind to the receptor sites. Therefore, nothing is being sent. There's no note. Remember, I was talking about the two people in class passing notes between one another? Well, now you can't pass a note from point A to point B. So since no notes are being passed to the muscle cell, that muscle cell is not receiving the command to contract. Therefore, there's no contractions happening. Now, to you, that may not matter much, but I just finished telling you about the skeletal muscles that help you breathe. So if the botulism, if those Clostridium botulinum toxins made it impossible for acetylcholine to cross the synaptic cleft and bind to the receptor sites on the motor end plate, then the muscles that are helping you breathe right now would stop. They would not contract. You would cease breathing, and you die in a matter of a couple of minutes. Not fun. Not fun at all. Okay, the last thing that I wanted to just chit-chat about was metabolic activity, and I'm going to skip on to the image here. And something you need to understand about muscle cells. I'm sorry, but muscle cells do not do all that work for free, bruh. It does not happen that way. In order for muscle cells to do what they do, they have got to have ATP. They've got to have energy. Now, ATP is a short-term energy. ATP in itself does not stick around for long. ATP is something that's supposed to be utilized right then. It's kind of like buying milk. Milk doesn't really last that long. That's why you keep it in the fridge and then you pull it out and you use it. As a matter of fact, let me use something that's a lot has a much shorter shelf life. Let's try an avocado. If you peel an avocado to use, you'll notice something real quick. Unless you put some lemon juice on that avocado, that sucker will turn brown the minute you cut it open and let it sit on the counter. You let an avocado sit on the counter, a fresh avocado sit on the counter for about 10 minutes, you look over there, all of a sudden it's starting to turn brown already. It's oxidizing. It's about to be mushy and nasty looking, and everybody's going to be like, ah, baby, don't use that avocado. That look nasty, baby. You don't want to use that. So you wind up putting lemon juice on there and crossing your fingers and hoping for the best. ATP is the same way, except there's no lemon juice to sprinkle on ATP. Once you make it, you better use it, because if you don't use it, you're going to lose it. So what we want to do is we want to utilize that energy as quickly as possible. But if we can't utilize that energy, then we want to store it. ATP is a taxi cab. Okay? ATP is a taxi cab. ATP in itself is just a molecule taxi cab. What's important to us is not the taxi cab. What's important to us is the passenger on the taxi cab. ATP happens to have 3 phosphates attached to it. That third P, that third phosphate, he is connected to the rest of the taxi cab via a high energy bond. In other words, we forced a bunch of energy into that bond that's attaching that third P to the taxi cab. So when we take ATP somewhere and we snatch that third P off, that releases the energy that was trapped in that bond, and our cells have the unique capability of harnessing that energy as fuel. It is one of the most amazing attributes of microanatomy that you could ever watch. A cellular respiration and cellular metabolism. It is absolutely amazing. It's absolutely fascinating, because you can look at this, and just the fact that you can create that small scale energy and harness it on a cellular level, that is amazing. You may not think that's that amazing, but several decades ago, a group of scientists figured out a way of snatching small micro orbital substances off of their rings, and they created this thing called an atom bomb. Yeah, you might know about that, all right? And so when you separate a substance from another substance that's being held together by energy, you release that energy, and some of it is going to be lost, and the rest of it can actually be harnessed and utilized. The same thing happens here. Every time your cell uses ATP, a large chunk of that energy that is released by pulling that third P off, a large chunk of that energy is lost. You don't actually use it. It's actually lost in the form of heat. That's how we stay warm. But the rest of that energy that we keep, the other 60%, that we actually get to keep, we utilize this energy, and it helps our cells to do its daily, day-to-day things. I mean, it relies on ATP like you rely on water and air. I'm serious. If you take ATP away from a cell, that cell will die. If you take ATP from a muscle cell, your muscle cells will shut down. They will begin to cramp. They'll shut down, and you'll be sitting there with a Charlie horse crying on the side of the road. So you want your cells to have ATP. I think I read somewhere that if I was sitting down, which I'm actually sitting down right now, but if I were to stand up, I read that it's going to take my muscle cells 25, what is it? I got to remember how it said it. It said that while I'm sitting here, Each one of my muscle cells, my skeletal muscle cells, each myofibril has like 25,000 ATP molecules ready to help me contract, to help me stand up. But once I start walking, that stored ATP that was just sitting there doing nothing, I run out of that. I burn right through that immediately, and then the rest of the way, I'm on my own. Literally, at that point in time, I have to come up with the rest of the ATP. It's kind of like, paying for something. It's kind of like going on a trip. If I go on a trip and I've got $50, that $50 may get me, and I'm going on a trip for a week. That $50 may last me a certain number of days, but then after that $50 is up, I got to come up with another way to come up with some more dough. And so that's where you're sitting at that moment in time. So what we have here is our muscle cells have a way of producing ATP in order to produce enough energy for your muscle cells to continue contracting beyond just an immediate movement. And so we have what's known as short-term energy supply and long-term energy supply. Short-term energy supply is anaerobic in nature. It happens out in the cytoplasm or the sarcoplasm of the muscle cell. It does not require oxygen, henceforth the name anaerobic. Anytime you have a or an in front of a word like that, that means without. So aerobic means oxygen, or with oxygen. So anaerobic means without oxygen. This process, known as glycolysis, which is the short-term energy supply process happening out here in the sarcoplasm, this glycolysis basically takes glucose molecules, sugar, and it runs it through a multi-step process, which is not shown here, but it runs it through a multi-step process to create 2 ATP molecules and 2 pyruvate molecules. These 2 pyruvate molecules are then combined with oxygen and transported into the mitochondrion of the skeletal muscle cell to undergo long-term energy supply making, which is known as aerobic cellular respiration. Notice that it's aerobic because we add oxygen to it, and the oxygen and the pyruvate go into the mitochondrion, and then it goes through a series of processes known as the citric acid cycle, which ultimately gives us numerous ATP molecules. And these numerous ATP molecules are then either utilized at that very moment, or the ATP molecules can then be taken on to have their third P's removed and have that energy stored as hold on a second, creatine phosphate. I was thinking about taking you to that slide, but I'm running out of time here. So that's when you go back in your notes and you see it talking about CP, creatine phosphate. That's when you take that ATP, you take that third P off of the ATP, which is the passenger in the cab, in the taxi cab. That third passenger takes himself and the energy and goes over to the creatine, and then attaches itself to creatine, and then this becomes creatine phosphate. And what creatine phosphate does is it basically takes lemon juice, pours it on that avocado, puts it in a Ziploc bag, and puts it in a special storage facility in your high acidity $3,000 refrigerator in the kitchen, which is going to store that avocado for quite some time so that you can pull that energy out and use it later. Because maybe I don't want to use that ATP right now. I mean, in a little while, I'm going to get ready to go down and fix a sandwich and some chips, all right? That's not going to happen for the next few minutes. So until that actually happens, what do I want to do with that ATP? Well, store it so I can use it later. Why spend it when you don't have to spend it? Just store it so we can use it later on. And that's really what you want to do. And that's why we're looking at long-term energy supply, because long-term energy supply lasts, that's for energy for any activity that's going to last longer than a few seconds. Longer than like 7 to 20 seconds. You know, this is like walking down some stairs, walking up some stairs, walking to your next classroom, walking to your car in the parking lot, walking over and asking that girl for her phone number. Oh, wait, I'm calling somebody out, ain't I? Or, you know, anyway, it's what you do aside from sitting there, sitting still, like short-term energy supply. And you know what? I think that's all we're going to talk about for right now, because I said I wanted to keep this really short and sweet. So we're going to go ahead and wrap this up. Look forward to talking to you guys soon enough. We will be talking possibly about some more material. Be looking out for some stuff on neurophysiology. We're going to talk about the nervous tissue. We'll talk about the different neurons. We'll talk about how does a neuron make an action potential in the first place. That's going to be a good topic to get into. So this is the professor signing off. Peace.